So, hello friends, welcome to my channel. And in molecular biochemistry made simplified, we have completed the up to pyrimidine synthesis, and now we are starting with DNA replication and RNA replication transcription and translation. Sorry. So let's start with DNA replication flashcard. You can see my three videos which I had made earlier. Now in this continuing the series, I'm continuing with DNA replication flashcard. Side by side, I will also make some more videos of the most important points of INI-CET and NEET PG, which are to be remembered till the last day of exam. So stay tuned with me. I will make side by side more videos in other topics also, which should be covered for INI-CET, seeing that 37 days are remaining for INI-CET. And so let's quickly start the DNA replication flashcards. Let me clear to you that these points are must know, and if you know these points, then any MCQ will be very quick for you. You can solve any MCQs if you know these points. In exam point of view. Okay, so this is concise, crisp flashcard, which are made for your last revision and last point revision, and your daily revision also. So let's start with DNA replication. As we know, this is occurs in S phase of the cell cycle and semi-conservative. That is, daughter strand are made from each parent strands and each standard template. So if there is an error, it is highly clinically oriented. Okay, so you will be able to solve all medicine questions also if you know these biochemistry topics very thoroughly. Okay, so mutation cancer occur due to its defects. Okay, so we will learn in example also later. Origin is at 80 rich sequence, that is adenine thymine. Okay, 80 rich sequences are origin of replication. Okay, helicase unwinds. Okay, it opens the DNA strands for replication. So 80 bond open easily, that's a mnemonic. On helicase, if there is a helicase defect, the DNA are not going to be opened, and it leads to Bloom syndrome. Okay, Bloom syndrome. We also know that progeria, that is also due to this. This is Bloom syndrome. Okay, early aging, premature aging is that. Okay, so DNA replication, it's a short diagram for your understanding. RNA primer just bind to it, and primase. What does primase do? Primase just give the template to this DNA. Okay, RNA template is given by this primase, and this is removed by this DNA polymerase. Okay, DNA polymerase 3 goes forwards and make in 5 to 3 direction. Okay, 5 to 3 direction, and this uses this primase. Okay, our main enzyme is DNA polymerase 3. Okay, and and what is the function of 1 and 2, guys? What is the function of 1 and 2? Just think about in your mind, and just try to recall. Recalling is the key, guys. If you in MCQ, there are so many options, and you will get confused. What is 1 doing? What is 2 doing? What is 3 doing? So basically, that DNA polymerase 3 is the main enzyme which leads to replication. Okay, so DNA polymerase 3 is the main enzyme, and everyone proofreads here. Okay, proofreading is done in 3 to 5 direction. Okay, always reverse. Okay, and this replicates in 5 to 3 direction. So this is the core thing we need to know. Okay, and now the DNA polymerase 1, and listen, guys, this is only in prokaryotes. Okay, this is going in prokaryotes. In eukaryote, there is alpha, beta, epsilon. This is all whole thing in DNA. But in prokaryote, there is 1 to 3 only. Okay, 1 to 3. So synthesis occurs in 5 to direction, and proofreading occurs in 3 to 5 direction. Okay, so 2 does this, and 1, that DNA polymerase 1, that removes our primase. Okay, which earlier we have attached to it. Okay, so this is the whole sequence of this thing. And one thing also we need to remember, this topoisomerase, this stabilizes it. Okay, topoisomerase, the supercoils which are formed in backward direction due to opening this topoisomerase, this remove this tension. Okay, this remove this tension which occur due to that wiring effects. Okay, and next helicase we know that it opens and ligase joins the Okazaki fragments. Okay, this lagging strands. This is joined by DNA ligase. So this is all the whole crux of this DNA and mutation. We have to learn the most clinical things. Okay, if you forget any steps, it's okay, but you don't have to forget any step which is leading to a disease. Okay, so this point mutations are like silent, missense, nonsense. Silent, let me clear you, silent is nothing but just it is the three sequence na, A C G A U G. So this third is like we are just reading in a hurry. Okay, so this can be skipped, but amino acid is made same. Okay, similar amino acid made, so this is silent, there is no need if it is A U anything else, but we have read that in sequence, so this is silent because in reality there is a change in something. Okay, change is going on a codon, but we are making the same amino acid, so this is silent. Okay, missense means like in sickle cell anemia, valine welcomes and glutamine goes, so in place of G there is V, so this is missense है ना, G की जगह V आ गया. Nonsense means stop the nonsense, मतलब stop codon आ गया. ठीक है, AUG UGA. Okay, UAA, this comes, if this comes then this becomes nonsense. So you can learn by silent is nothing happens, nothing happened in general, but nonsense means stop codon has came, and missense mean altogether changed the codon. Okay, glutamine in place of glutamine valine comes, so sickle cell anemia. And what's the example of nonsense mutations, guys? Nonsense and missense, we have to learn this. Okay, so in the particular nonsense mutations is like terminal protein. Okay, we have made the terminal protein, there's a truncated protein formed like in our DMD है ना, Duchenne muscular dystrophy. And also in DMD also frame shift also happens. ठीक है, frame shift means any codon in group of codons is happened. Okay, like addition of any codon or deletion of any full codon, full 3 set codon is lost or it is added, so it changes the whole reading of the frame, guys. Okay, so this example is DMD. Okay, Duchenne muscular is a motor disease and over sign positive is there in Duchenne muscular dystrophy. There is an axial hypotonia and child is so floppy, so it's like due to this mutation that happens. Okay, and its severe form is DMD and is just partial deficiency, partial protein is formed in Becker's. Okay, they are all X-linked recessive. So this is the example of frame shift. And nonsense can also happen in DMD and formation of truncated protein, like less protein. Okay, because earlier it stopped in earlier it has stopped, so that forms a truncated protein. So expansion mutation like trinucleotide repeats, CAG repeats in our myotonic dystrophy, CTG repeats in our myotonic dystrophy, and our CAG repeats in Huntington disease, trinucleotide repeat disorder, Fragile X, and everything like it goes in expansion, continuous repetitions of this protein है ना, so happens in this Huntington's chorea. So Huntington disease. Okay, sorry. Clinical poll. So this is sickle cell. This is our missense mutation. Okay, cystic fibrosis. In cystic fibrosis, what has happened? Like alteration of any transport protein, like delta F08. Now this is a transport protein, and this can happen due to any frame shift mutation or a nonsense mutation, just like same like DMD. So cystic fibrosis is also a most important thing. And one thing also you should remember, later I will tell in RNA transcription that alternative splicing occurs in cystic fibrosis also, so different protein is formed also. So cystic fibrosis has so many pathophysiology, and we have to understand in core. Okay, AIIMS love this cystic fibrosis topic. Okay, so frame shift mutation can happen in cystic fibrosis. Okay, so you should understand this. Okay, so frame shift mutation. And one more example is Tay-Sachs disease. Okay, Tay-Sachs, so you should learn this. Okay, Tay-Sachs disease, in which we have cherry red spot, no hepatomegaly, and no hepatomegaly, tall stature, startle reflex is positive. If you clap in front of child, it will give no response. Okay, no, sorry, it will give exaggerated response. Okay, when you clap in front of a child, so that is startle reflex exaggerated, and that's in Tay-Sachs. Tay-Sachs can be differentiated from Niemann-Pick diseases in presence of no hepatomegaly in Tay-Sachs, and this can be differentiated in from Gaucher, in that Gaucher has no cherry red spot. Okay, it can be present, it cannot be present, but Tay-Sachs disease is an example, cystic fibrosis is an example, and DMD is an example, and nonsense can also occur in these two. Okay, so I have told you clinical pulse in this, and now let's move on to the same thing. This is the example, X-linked recessive nonsense mutation. Tay-Sachs has also a hexagene mutation. You should remember that. They can ask anything related to that. It can also come in NEET-PG nowadays, because they are also loving the first second profs nowadays. So frame shift mutation, point mutation in GM2 accumulation. So T, you should remember T, na? T2 sounds like T, because GM1 is cholera. Okay, cholera toxin, but GM2 is Tay-Sachs. 2 से Tay-Sachs, TT is sound similar, so this has come into this year's NEET PG, Tay-Sachs also, hexosa aminodase A. Okay, Tay-Sachs. So this is the enzyme deficient. Okay, so this is accumulated protein because of DNA mutation defect. So muscles down and a severe genetic disorder they will cause. So let's move to RNA transcription. Guys, RNA transcription in eukaryotes, they are mainly from RNA polymerase 1, 2, 3. Reverse like DNA. Okay, DNA transcription has prokaryotes 1, 2, 3. But in prokaryotes, this is a single RNA polymerase doing all thing. Okay, all thing. It has single RNA polymerase, but it has 1, 2, 3. One codes for rRNA, two codes for mRNA, and three codes for tRNA, most important. Okay, and this 2 is also causing 5 RNA sequence. Okay, so you can remember by RMT. Okay, 1, 2, and 3. This polymerase does this rRNA, mRNA, and tRNA. Defect in this 2 RNA will cause amantadine, caused by amantadine. Amantadine inhibits this mRNA. Okay, and this will cause hepatotoxicity and on this comes in mushroom. Okay, mushroom poisoning. And 3, and we know that dactinomycin, which is an antibiotic, this inhibit both prokaryote and eukaryote RNA polymerase. So this is the most important question. Okay, this inhibits both. And in prokaryotes, which inhibits the RNA polymerase? Rifabutin, and which we use in our HIV, and which is used in our rifampicin, our TB drugs. Okay, HIV TB drugs, and this rifampicin inhibit DNA dependent RNA polymerase. Doctor, you should remember doctor. Okay, DNA dependent, DNA comes first, then RNA. Okay, DNA dependent RNA inhibited by rifabutin and rifampicin in prokaryotes. Okay, in eukaryotes I told amantadine, this inhibit 2 RNA polymerase 2, and dactinomycin inhibits both. Okay, RNA, and everything. So this is the clinical pearl. I have told transcribe to RNA. Error in transcription also causes diseases. Okay, cytoplasm occurs in nucleus in eukaryote and cytoplasm in prokaryotes. So promoter region. First, we have to bind this RNA polymerase, this enzyme, to our promoter region. Okay, and this is a enzyme rich box. Okay, Tata box in eukaryotes. Eukaryotes had a Tata box. This is initiation factor always binds to this, then transcription start. Okay, and this mutation, the transcription protein binding mutations affect transcription initiation. If there is a defect in this Tata box and initiation process, then also we get some diseases. So let's see next, if and I have told in very short, but this is a big process. Okay, big process, but in exam point of view, this transcription process has no such importance unless and until it is asked in a way of clinical importance, like in sense of any diseases. Okay, so we have to learn that diseases, and we have to learn our splicing mutations. Okay, splicing mutations in first aid also, it has given only up to that which I told you. Okay, 1, 2, 3 RNA polymerase, their functions pass only that, but the main thing is this splicing. Okay, you have to know this splicing process, because this has many genetic diseases. Because this, how is this spliceosome is formed? We know that intron is the heterochromatin part. It is of no use, and that dark RNA, we have to remove this or make this exons join. So this process, if these are joined in a such a way that the snRNA, small nuclear RNA, is that, okay, this is hetero RNA, so this is called small nuclear RNA. And also protein made by this is called snRP, okay, small nuclear, because this is heterogeneous. Okay, till now we are in stage of pre-mRNA, think like that. Okay, no mature RNA is formed till now. And how this is removed? An snRP is binded with this, okay, this intron, है ना? And a ribosome also, okay, ribosome complex is made, okay, 5 splice site. So this imagine this is 5 site of intron, and this is 3 site of intron, okay, because this is 5 3, and this snRP has to bind with this. And now this take this intron with this, and snRP should go out and make a spliceosome in form of lariat. Okay, this is intron, and this is 5 3. Just we have to take this part, okay, in like, and so this is form a lariat shape, and this has taken out of this, and now exons are joined. Okay, 5 3 continuity. So this process, if it is defective, then this is called splice site mutation. Okay, splice site mutation occurs in sickle cell anemia. So you have to listen all the disease which I am telling and their pathophysiology here only, so that when you go to medicine and you learn this, you understand why it has happened. Okay, so biochemistry is not a such a paraclinical or non-clinical subject. It's now, according to our AIIMS exam also, it is so important, very important for clinical diagnosis. So this is all written here. Okay, intron is cut at 5 to 3, and intron excised in the lariat form, and the remaining exons are joined. And one thing also I will like to talk here only, that alternative splicing is a term frequently used in AIIMS question exams. Okay, option, as an option. This is what, this is like we can make multiple proteins with a single pre-mRNA. How? Because this introns, suppose I change the color, this introns, this can be taken out in any sequence. Okay, this can be taken out like this and this can be taken by this, and in this two can be joined together, or this two can be joined together, like 1 2 3 4. So any sequence they can get joined. If the sequence is changed, like this is the sequence, okay, 5 3 5, like this is the sequence, you think like that. So this part is taken out, this is one. Oh, sorry, sequence is always same, 5 to 3 direction. This is the intron. Okay, this is the intron. So like this has been taken out, but this part, this part, this part, this part, this is all exon. Okay, so maybe one is joined with 3, or 2 is joined with 3, taken out. So this all protein will make something different. Like in brain has different dopamine receptors. So this is clinically important. Okay, why the different dopamine are formed by same amino acid, same proteins, how they are made, because of alternative splicing is going on, है ना? Like in cystic fibrosis also different protein is formed, or tropomyosin in muscle also different. This all is given in first aid guides, most important. So these are so hotshot topic of examiners. So muscles, there is different type of tropomyosin in muscles because of this alternative splicing. Okay, so this is different. This is completely splicing process. But alternative splicing, this can occur like this: one can join with 3, or one can join with 2. Okay, or 3 can join with 2. So these are all make and different protein. Introns are removed. Okay, there is no problem in removing introns, but in joining this happens in different variations. So this occurs. Okay, different tropomyosin protein in brain, different dopamine receptors are formed. Okay, and in tumor cells also different defense mechanism are adopted due to this alternative splicing. Okay, so this is alternative splicing, you should remember this. And mutation of splicing can lead to disease like spinomuscular atrophy. क्या है? This SNRP protein is defective. This is not able to join this and form a lariat. So this leads to anterior horn cell defect. Okay, anterior horn cell defect protein is defective, and this leads to floppy baby syndrome. Symmetrical weakness occurs in SMA. Okay, and you have to remember everything. SMA has many new drugs coming on, so you have to learn also SMA new in further course. I will tell also that also, because I have recorded that also. I have made the flashcard of most important diseases asked by AIIMS examiner repeatedly in every exam. They can't take any exam because without asking these things. Okay, so I have recorded that one, and I will show it to you. So this spliceosome formation is defective in SMA also, and this led to floppy baby syndrome, anterior weakness, symmetrical weakness, and also there is also a disease called as SLE. You know that so many antibodies are involved in SLE. What is that? The Smith antibody requires this SNRP protein also. Okay, if this is defective, they join with Smith protein, Smith. Okay, Smith antibody is formed, anti-Smith antibody is formed in SLE, and this is defective in also splicing. So this SLE is also important by this point of view. Okay, this is a splicing defect, we can say. Anti-UNRP protein, ribonucleotide protein, is defective in mixed connective tissue disorder like dermatomyositis, polymyositis. All this thing is related to splicing. Okay, this all are required for this process. Okay, this intron removal process. Thus understand this, and you are sorted. Okay, so and what diseases I have told you, again revise: SMA, spinomuscular atrophy, SLE, and mixed connective tissue. What are the antibodies? Anti-Smith antibody, okay, anti-SNRP antibodies, or anti-U1 SNRP. So this is all the crux of this. Okay,